Chapter 12, Biochem 2, Fat-Soluble Vitamins. So a lot like Chapter 11, there's going to be a lot of just kind of general information in here. A good chunk of reading. More than likely, I'm probably going to skip a good chunk, just the random stuff, and focus more on the stuff that you're going to be tested on. Because while there is a lot kind of in here, there's not that much that's really what you need to know. So things that we're going to look at is the functions of the fat-soluble vitamins, differences and similarities of the fat-soluble and water-soluble vitamins, the food sources, the mechanism of action of the vitamins, so kind of what they do, where they go, what they're acting on, and then signs and symptoms for deficiencies and toxicities. So the ones that we remember, it's A, D, E, K. So kind of the way to remember this is kind of, it's a little vulgar, a little stupid, but if you remember A, fat, dec, that's the easiest way to remember those fat-soluble vitamins, A, D, E, K. So characteristics, they are apolar hydrophobic molecules. Hydrophobic, they're fats. Fats don't mix with water. And they are all derivatives of the isoprene units. Think way, way back to when we were talking about fatty acid biosynthesis, how, you kind of, before you actually make the full fatty acid chains, you make those isoprene units. So same thing here, they're made out of isoprenes. These cannot be synthesized by the body in adequate amounts, so they must be dietary and absorbed by the GI tract in the same way as any dietary fats. So think of these the same as like our linoleic acid and those essential fatty acids that we need to consume them. Absorption of lipid-soluble vitamins requires normal fat absorption, so they go with the normal fat. They're absorbed along with the dietary fat. Bile salts, lipoprotein, lipase are required. Remember, the bile salts coming from the gallbladder, the lipoprotein, lipase coming from the pancreas, right? Or spleen. Sorry, brain fart with that. Should be the pancreas. Yeah, from the pancreas. Sorry about that. Little brain fart moment. The lipase is the enzyme that breaks down fat. Any condition with the diseases of the biliary system and anything that interferes with fat absorption can lead to malabsorption of the fat-soluble vitamins. So start to think of things involved. So think gallbladder issues, think liver issues, think pancreatic issues, think intestinal issues, leaky gut, Crohn's, all that kind of stuff, inflammation of the gut. Anything that's involved within all of those parts are going to be responsible if we have an issue with absorbing it. Transportation of those, so they're transported to the liver by those lipoproteins, the chylomicrons, same as with fats. A lot of kind of repeat information here. In blood, the vitamins are transported by lipoproteins or specific binding proteins since they are not soluble in water. Liver is the major storage and repackaging site. Only kind of exception is like long-term storage. Vitamin E is more in adipose tissue, whereas the other ones, A, D, and K, are more in the liver. These kind of things, these will also be questions in physiology 2, so you'll want to remember a lot of these stuff because these are going to come back. Excretion, the lipid-soluble vitamins are not excreted in the urine, but are more likely to appear in bile and thus excreted in feces. Toxicity, because of the fat solubility of these fat-soluble vitamins, they can produce toxicity if stored in excess amounts. Vitamin supplements do not exceed 2 times the RDA safe. So unlike the water-soluble vitamins, which you can essentially take as much as you want because you're just going to filter and pee them out, do not do that with the fat-soluble vitamins. Most common ones to overdose, vitamin A and vitamin D or for overdosing. Biomedical importance. So this is going to give you kind of the overall, hey, these are the important things that you want to remember. So with vitamin A, whenever you think about vitamin A, I want you to think about first the eyes, and then you can also think about the skin. Mostly the eyes though. So if we have a vitamin A deficiency, we can have night blindness, and then we could also have xerophthalmia. Xerophthalmia is kind of what's the word for it? Kind of like an overarching term. It refers to it calls it a spectrum of ocular diseases. So basically like a whole bunch of different eye diseases will be called xerophthalmia. So this kind of ophthalmia right there, that is Q for our eyes. If we ever see that suffix, we're thinking about the eyes. So basically anything with the eyes is going to be vitamin A type related. Vitamin D deficiency. So vitamin D, super, super important. We need it for bone building. So a lot of times, like things like rickets, osteomalacia, this is either a vitamin D / a calcium issue. So if we don't have enough vitamin D or we don't have enough calcium, then we get issues. These present differently. I don't think you guys have reviewed these kind of stuff yet, these diseases in your radiology courses. If not, you will see them. Rickets versus osteomalacia. Just remember, rickets is for children, osteomalacia is for adults. Vitamin E deficiency, we have some neurologic issues as well as anemia. Vitamin K deficiency, bleeding, and hemorrhage. Think way back to our electron transport chain. And remember how we have that enzyme, that coenzyme Q, that CoQ10. Remember, it looks very similar to our vitamin K. We need that vitamin K for CoQ10. So basically, if we have vitamin K deficiency, we can't produce ATP. Non-production of ATP, cells start to die, and our whole body starts to fall apart. So that's vitamin K. Remember, think electron transport chain, that coenzyme Q, CoQ10. Vitamin A, there's going to be a whole bunch of different names that you're going to see for these vitamins, and a lot of it is just going to be kind of putting those names together all into one group so that you can know how, when you get asked a test question, sometimes instead of saying vitamin A, they're going to say, oh, it's a deficiency in retinol, a deficiency in retinol, deficiency in beta carotene, that kind of stuff. So they're going to kind of try and trick you in that way when they ask questions about this. So again, deficiency, blindness in the world. Deficiency of vitamin A is the leading cause of blindness in the world. Lack of vitamin A causes dryness of eyes. In poor countries, associated with infection and premature death of children. Of course, you have the generic vitamin A. Here are all of the different names. They're called retinoids. Retinoids, retina is in the eye. Remember that. So retinol, retinal, retinoic acid, and then vitamin A exists as beta carotene. Everybody knows beta carotene. Think about carrots is the big source of beta carotene. And beta carotene also is technically provitamin A and a bunch of other types of carotene type stuff. So these are the ones that you're going to want to remember. Big ones, key ones, retinol. So this is that full vitamin A activity. So this is like our most active. Beta carotene can be converted up into other stuff that we need. Okay, so big sources of food. Plants. Big one is going to be carrots. And then the other one that you want to really remember is liver. And this is the other note: avoid liver of saltwater fish and marine mammals. Not necessarily like, don't eat it at all, but you have to be very, very careful with how much you consume, because liver is because remember, A, D, and K are stored primarily in the liver in mammals. So if you eat a bunch of liver of animals, you're getting a massive dose of vitamin A. And vitamin A is one that, can cause some issues. There's actually a story, I don't know if he still shares this story or not. There's a story of a ship, an explorer ship that went to explore Antarctica, and essentially what happened was they got stranded. Their ship got stranded. This is 250 something years ago, where it was still like wooden ships, and so they got stranded there, and they ended up killing a polar bear and eating the liver of the polar bear, and a lot of the crew basically went nuts, and like a whole bunch of other craziness ensued from that. So you just got to be careful with how much liver you're eating. Let me see if it gives us any toxicity. Okay, well, I'll talk about that when we get down there. The conversion of vitamin A forms retinol can go to retinal, and retinal can go to retinoic acid. Retinol and retinal are interconverted in the presence of NAD or NADP, dependent dehydrogenases or reductases present in many tissues, so these two can go back and forth. Once formed from retinol, retinoic acid cannot be converted back to retinol or to retinol. Absorption and digestion. Retinol esters are dissolved in animal fats. They disperse in bile droplets to produce what are called micelles, hydrolyzed in the intestinal lumen by hydrolase to form free retinol. And free retinol is directly absorbed by the intestinal epithelial cell. So this step, this process, you're going to want to remember how you have these retinol esters, these little esters being like the ester bonds. Adding bile salts, they make micelles. Hydrolases turn it into free retinol, and it gets absorbed. Beta carotene is similar. Beta carotene from like those carrots and stuff. You add bile salts and oxygen to it, it turns into retinal, and then it can go to retinoic acid, or it can be turned into retinol. And again, those beta carotenes are present in plants. Retinol is reduced to retinol, retinol reductase. Underlined, that'll probably be just a specific test question. Retinal is reduced to retinol by retinal reductase utilizing NADPH. A small fraction of retinal is oxidized to retinoic acid. Most of the retinol is esterified, so esterification being bound with saturated fatty acids, and incorporated into lymph chylomicrons. That's how it gets into the bloodstream. And then it goes to the livers. Into the blood, all the way to the liver. Here's just that whole process, kind of, again, laid out. Again, stored in the liver as a retinyl ester. This is where I recommend go and write down, have a little sheet off to the side, write down all of these names. So active forms: retinol, retinal, retinoic acid. Provitamin A equals beta carotene. It's essentially the same thing. We'll want to remember, write down retinyl ester is the storage form in the liver in lipocytes, aka adipose tissue, as lipoglycoprotein complex. So all of these are names. They're all saying the exact same thing. It's just in different forms. Okay. More of the same, released into blood and attached to binding proteins. Retinoic acid binds to albumin. Retinol binds to aporetinol binding protein. Retinol binds with cellular retinol binding protein, CRBP, inside extrahepatic tissue. So again, write down those names and what they bind to. Unique biological functions. Retinol supports reproduction. In reproduction, retinol acts like steroid hormones. It binds to nuclear protein receptors, controls certain gene expression. Immune function, important for activation and proliferation of lymphocytes. Retinol, needed for night and color vision in retina. Most of the times, this is what you'll be asked. Occasionally, they'll throw in immune function, and more rarely, they'll throw in this one up here. So, for sure know this one. This is a good number 2 to know, and occasionally this will be asked. This goes all... That's a little bit more in detail. I don't believe we really go into all of this, but we'll see when we get through line of sight questions. Component of the visual pigment rhodopsin, which is present in rod cells. Rod cells for black and white, or for rods are black and white. This is going to be a question he'll ask. Rods are black and white. Cones are color. All through there. That's what I just wrote right there. Bunch more stuff. It's kind of meh. This is what you'll want to remember. So rhodopsin, we'll want to write down what rhodopsin is. So rhodopsin is a component of the visual pigment in the rod cells. When it is exposed to light, it is dissociated as it is bleached, so as it is exposed to the light, and it forms all trans-retinal and opsin. This reaction is accompanied by conformational changes that induce a calcium channel in the membrane of the rod cell in a GTP-dependent manner. So GTP being the energy form that it needs. So calcium influx increases, which triggers nerve impulses, allowing light to be perceived by the brain. This is literally just talking kind of about the mechanism of how we see. Making it all easier, light enters the pupil, rhodopsin in rod cells get hit, it creates all trans-retinal and opsin. Because of that, it creates calcium channel to open. Calcium channel opens, ions move, and because of that, we get electrical impulse. Okay. I'm gonna read through this just in case we get questions on it and stuff. But it's gonna be a lot of kind of technical terminology. Don't try and understand all this right now. It's easier to get through with the help of looking at the questions. So role of cGMP, depolarization/hyperpolarization conditions, transducin proteins, the G protein in visual signal transduction. So the cGMP dependent phosphodiester enzyme. Again, when stuff like this comes up in questions, we'll look at it, but this is a lot of words right now that don't really mean a whole lot, so don't worry too much about this right now. More words. Transducin is the guanine nucleotide binding proteins required for visual signal transduction. Transducin contains 3 subunits, T alpha, T beta, and T gamma. In its inactive form, transducin binds GDP to its alpha subunit, A lot of this should sound familiar, this kind of stuff. Remember those G protein coupled reactions? Those G protein coupled receptors, those kind of stuff. Very similar to the alpha, beta, gamma components, all that kind of stuff. Binding of GTP to transducin alpha subunit activates the T alpha subunit, and as a result, T, beta, and gamma subunits are dissociated, exactly like G protein coupled receptors. The light absorption bleaches the rhodopsin. You get the retin, that should be retinal, and opsin, which converts the 11-cis retinal to all trans retinal. The pronunciation is weird because you want to differentiate AL from OL, but the signal from light absorption is immediately transmitted to inactive transducin, which is next activated by replacement of the GDP from its bound form to the active transducin, the T alpha, now bound to GTP. This is just exactly the same. All this is going over just a couple extra bits from the G protein coupled receptor reactions. It then activates the cGMP, the cyclic GMP dependent phosphodiesterase, by removing the inhibitor attached to PDE, making it active, and by converting cyclic GMP to GMP. In dark conditions, cyclic GMP levels are high, and the cell is depolarized by increased sodium ion / calcium ion influx, and in this condition there is no light perception and we cannot see. As soon as the cyclic GMP is broken by cyclic GMP dependent PDE, as soon as the cyclic GMP is broken by cyclic GMP dependent PDE to GMP, so we remove the cyclic part, cyclic GMP levels decreases and sodium calcium influx stops. Calcium moves out of the cell with a calcium sodium exchanger protein in the membrane, and this makes a hyperpolarized condition, and we can see so this whole process, there's a lot more detail in here that we really need to know. When we go through the questions, we'll come back to this, I'm sure, a couple little bits and point out what's important and stuff. So in general, think back to your G protein coupled receptors and kind of how those work, and we will have similar similarities between those two. Okay, continuing. Retinoic acid supports growth and differentiation, cellular differentiation and protein synthesis, like glycoprotein synthesis. Retinoyl phosphate is the carrier of oligosaccharides across the lipid bilayer. Remember what an oligosaccharide is. Saccharide, oligo being like a poly, having more. I believe it was more than 6 was an oligo. Differentiation and maintenance of epithelial cells. So that's where I said vitamin A does stuff with the skin and body surfaces. Lubricants, especially for the eye, prevent dry eyes. Xerophthalmia. I guess that's what they refer to that as, just dry eyes. Not just the only thing, but if you are looking at a question, if it mentions dry eyes, xerophthalmia is your answer choice. Also prevention of skin, lung, bladder, and breast cancer. All that stuff. Vitamin A deficiency conditions. Here's all of the potential ones. These are typically the things that get most tested on. Usually this will be second. They'll talk about immune depression, and then reduced mucus secretion occasionally, and then sometimes reproductive abilities. So the deficiencies, toxicities, because these are like the most important, I would throw these into flashcards and start reviewing these to recognize, okay, what which vitamin are each of these conditions correlated to from each of the different vitamins, including, water and fat soluble. Vitamin A toxicity. Large doses is highly toxic leading to cellular disruption and death. If the capacity of the retinol binding protein increases, it may cause accumulation of unbound retinols. As a result, cells are exposed to unbound retinols, which cause toxic effects to the cell. There is something else that occasionally gets asked more on boards than anything else. I believe it was vitamin A. I'm going to find it in a sec. The RDA for vitamin A. 6 micrograms, 12 micrograms. 1,000 RE for men and 800 RE for women. Let me see if this was... Okay, it was something else not related to vitamin A. Okay. So I wanted to make sure if there was something important that I made sure that I mentioned it. Okay. Yeah. And that is vitamin A. Let's see. We'll do a couple... How much is vitamin D? We'll do is we'll go ahead and stop the video here because we're just about approaching that 30-minute mark, and we'll continue in the second part with vitamin D. We'll see you in the next one.